Okay, so as Professor Buratti introduced to you, what we have been working for quite a long time now in the lab is this particular protein, TDP43, that we really discovered when we were looking for the transacting factors that were capable of affecting the recognition of CFTR exon 9. And really, talking about this protein, what I'm going to show you during this talk is the other side of the coin, as Professor Buratti talked. So Professor Buratti just told you that you have many mutations and how mutations in the RNA can affect the recruitment of transacting factors and therefore affect the recognition of this exon. What I'm going to show you really later on in the talk is how when you change a single transacting factor, how many splicing events can change and how these could be connected with disease. So at the time, of course, the only connection with disease between TDP43 and human disease was the cystic fibrosis. Totally unknown to us, in the same year, people who were working in the neurodegeneration field, they realized that when they started looking at the inclusions that were formed in the brain of patients affected by frontotemporal lobar degeneration, they could really divide the patients into two classes. One class where the inclusions were mostly made by the tau protein, and these patients carried mutations in the MAPT gene, and another class, much bigger than the tau one, or at least as many, that had ubiquitinated aggregates for which they could not actually name the protein that was responsible. And the answer to these questions, really, that changed our life completely, came in 2006. Because in 2006, these researchers from the US in Philadelphia, what they did was they managed to isolate the aggregates for which the composition was unknown. They immunized mice, obtained antibodies against the proteins that were found in the aggregates, and finally they managed to see what these antibodies were recognizing. And what they were recognizing, they didn't expect this at all, was TDP43 itself. So starting from 2006, the number of people who have been looking at how this protein can affect disease and especially neurodegeneration has dramatically increased. You see that all these, more or less, all these publications before 2006 and from 2001 come from Professor Buratti's lab. Now the publications have reached the level of approximately more than 200 every year, and they continue increasing, just to give you an idea of how important this protein has become. And of course, what is this protein? And as Professor Buratti said, this protein is a classical HNRMP protein. So HNRMP proteins, this is an old biochemical classification where initially when researchers were looking at the proteins that were associated with RNA, all the proteins that could bind directly with RNA were called HNRMP, for heterogeneous ribonucleoproteins. And heterogeneous ribonucleoproteins have, are very important for the entire life cycle of mRNA. As soon as the mRNA is transcribed from the DNA by the Pol2, the mRNA is never actually left as a naked molecule inside the nucleus. It is bound by a whole lot of HNRMP proteins, and these HNRMP proteins are responsible for the capping, the polyadenylation, the editing, the surveillance, the transport, and also eventually the translation of the protein. So TDP43, like many of these HNRMP proteins, what it does lots of things. Of course, it's not just a splicing factor like we found in CFTR, but it's also involved in microRNA processing, in the transport, the stability, and also the translation of of the messengers, which really means that when we look at the role that TDP43 is playing in the disease, we have to take into account that splicing may just be a facet, and of course we have to try and understand what is the role that the splicing process controlled by TDP43 may have with regards to the disease. As I said before, one of the characteristics of the disease is that this protein that is normally present in the nucleus and can shuttle from the nucleus to the cytoplasm is found specifically in the aggregates of the patients that are either cytoplasmic or nuclear. And in these aggregates, TDP43 is not only in an aggregated form, but is also cleaved, it is phosphorylated, and it is also ubiquitinated. Now, there are several ways through which this aggregation can occur, and of course there are also several ways through which this aggregation can be reversed, and the one that we are going to focus in this talk we really have to do with the autoregulation and the interaction with other proteins, because as you can well imagine, the interaction with other proteins can actually help keep the protein soluble, and also if you have defects in the production of this protein, you could have an increase in the formation of the neuronal aggregates. And starting from how important is the interaction of proteins with regards to aggregation, we really built up on an observation we did in 2009, where using a fairly simple far western technique to see what proteins were bound preferentially by TDP43, when TDP43 was used as a probe just to hybridize with the Western blot, what we found is that TDP43, like many HNRMP proteins, like to bind other HNRMP factors. And we then started, of course, to see if this binding was functionally relevant, and what we did here is we adapted the minigene technology that Professor Barale just explained to you, so we used the CFTR exon 9 minigene, where in normal conditions we have some exon inclusion, but also some exon skipping. We removed the TDP43 that is binding to the UG repeats in front of the 3' splice site, and as expected, you see that there is no exon skipping anymore. Now CFTR exon 9 is efficiently recognized. Then we made we transfected back into the cells an SI-resistant version of TDP43. This is a positive control, so we actually proved that if we put back TDP43, then we recovered completely the exon skipping. And then we removed the sequences in the C-terminal tail of TDP43 that we thought were responsible of binding to HNRMP proteins. Now, as you can see here, putting back a TDP43 that didn't have the C-terminal tail, we could not manage to recover the exon inhibition, and then I won't go into show you the data, and then using selective deletions in the C-terminus, we ended up with a sequence that goes from residues 321 to 366, that when we deleted, we also could not recover the skipping ability, and this deletion was associated with an inability of binding of TDP43 to these HNRMP factors. So this was the proof that, at least functionally, the interaction between TDP43 and these other HNRMP proteins that belong to the AB family is actually relevant for splicing. But we also noticed one thing, because whilst we were basically validating the binding data, so what we were doing was just simply do a band shift analysis. So you have this is a UG repeated probe. When you bind TDP43, you have a band shift because the complex is much bigger. If to this UG TDP43 complex you add HNRMPA2, that is one of the HNRMPs binding to it, then you can see the formation of a supershift. And what we were interested in doing to validate the binding was to look to try and incubate also this complex here with a peptide that was carrying the region that we had just mapped as functional. And you can see that indeed just adding the peptide from the C-terminus domain of TDP43, this action could abolish formation of the supershift because the peptide was binding to the HNRMP proteins and therefore removing it from the binding reaction. But not only we abolished the supershift, but we also started seeing the formation of an aggregate or of a very retarded complex that was evidently of very high molecular weight and was migrating and was actually left up in the wells of the gel. And so this started giving us a clue that interaction of HNRMP proteins was not just due to the presence of this particular sequence in the C-terminus of TDP43, but it could also influence the solubility of the protein. And this sequence was particularly interesting from a compositional point of view as well, because it was a sequence that was fairly, this 342, 366 sequence was a sequence that was fairly rich in QN polyglutamine repeats. And these QN polyglutamine repeats are fairly famous as far as protein aggregation in neurodegenerative diseases are concerned, because they can act as an aggregation focus. So when you have these sequences, the protein tend to actually aggregate. Here you see in a filter binding assay. And what is even more important, when you have this aggregation, the aggregation can act as a protein sink. So it will act as a trap for your TDP, for the protein itself. And this, in this case of TDP43, was shown that it could actually influence the splicing process. So if this sequence is important, what we tried to do approximately a few months ago was to set up a system to exploit this particular region of TDP43 and try and reproduce the formation of the aggregates in culture cells. The way we did this was to simply use a reporter protein, that is a classical EGFP protein, and we added to this reporter protein a increasing number of these 342, 366 repeats. And we then, first of all, tested what was the aggregation ability of these proteins using filter binding, and you can see here that if you add just one repeat to the EGFP, you practically have no aggregation whatsoever. If you add 4 repeats, then you start seeing some aggregation. This is a filter binding assay. But if you have 12 of these repeats, one after the other, then the protein aggregates very efficiently. This EGFP 12 per repeat. And what I think is even more interestingly is that if you transfect this effector, let's call it this way, into U2OS cells, and you look at the endogenous, or the amount of soluble TDP43 that is made by these cells, you can see that when in normal conditions, when you just add EGFP, the endogenous TDP43 is evenly distributed between the soluble and the pellet fractions. But if you, to these cells, if you transfect the 12 times EGFP protein, then the shift from the soluble to the pellet is rather dramatically affected. And so this gives an idea that if you put this effector into cells, you really reduce also, not only you form aggregates that of course will contain this effector, but you also dramatically reduce the amount of soluble TDP-43. And what we did then, of course, beside these solubility assays, what we looked to see if in these cells there was the formation of the aggregates, and indeed, as you can see here, we saw that there was a rather nice formation of several dots in the cytoplasm that could colocalize both with the AGFP staining and the staining for TDP, for the endogenous and full TDP-43 protein. And you can see that, and what is more interesting is that if you, instead of looking at the endogenous TDP-43 protein, you transfect a protein that lacks, the TDP-43 protein that lacks the 342, 366 region, then of course the recruitment of this protein that lacks the region does not occur anymore. So this was actually a demonstration that through the region, the 342, 366 region in the endogenous protein is actually necessary for it to be recruited in the aggregates. The aggregates were also both ubiquitinated and phosphorylated, and this is very good news because we know that the aggregates in the patients have these two properties. And this effector not only worked when we looked at the culture cells, so U2OS and normal culture cells, but it also worked when it was transfected into primary neuronal cells from rat. So it is something that we could, also in a neuronal setting, we could reproduce the formation of these aggregates. And so the idea that we had and that we concluded in just recently is the fact that, of course, there are many ways that can lead to TDP-43 aggregate formation, but most probably the interaction of TDP-43 with other HNRMP proteins is something that is quite important to prevent this process from starting and eventually growing. Now, what is the consequences of such a process? And here we get very near to the splicing story of TDP-43 again. Well, one possibility is that the aggregates themselves are toxic, but at least in our system, the formation of these aggregates using this effector didn't show any toxicity whatsoever. These are positive controls. This is a toxicity assay in our cells. And so one possibility is that the aggregates really are, by themselves, are neutral. So the formation of the aggregates inside the cytoplasm is not very harmful for the cell. What could be very harmful for the cell is the fact that these aggregates are recruiting the endogenous TDP-43 and they are depleting the endogenous protein from the nucleus. If they deplete the endogenous protein from the nucleus, then you can well imagine that all the mechanisms that are controlled by TDP-43 in the nucleus will become disrupted, and I'll show you in a minute how. Before we do that, just let me give you very briefly, when we wanted to look at these kind of issues, what we started to do was to make stable cell lines. So we made stable cell lines that were all capable of expressing a transgene of TDP-43 that was SI resistant, and the idea was that using these cell lines we could deplete the endogenous TDP-43 and then switch on the expression of the transgene. And of course, not only the transgene in its wild-type conformation, but we also wanted to express, to see what was a possible consequence, the sequences of TDP-43 that lacked, for example, the RNA binding ability, or just the sequence that was responsible for HNRMP binding, and see what were the effects. And immediately, as we were kind of just looking if the system was working, what we did make was a very interesting observation. And that is, when we overexpressed the transgene for the wild-type TDP-43, even without removing the endogenous protein, using an siRNA reagent, what we saw was that the endogenous protein was downregulated. And this did not occur when we inserted a mutant, when we induced the production of a mutant TDP-43 protein that was not able to bind RNA. So obviously there was an autoregulation mechanism that was going on, and that was dependent on the ability of TDP-43 to bind RNA. The most likely candidate of binding of TDP-43 to its own RNA came from a particular kind of analysis that I'm not going to go into it. It's called CLIP. But basically what this analysis gave us, and that we validated using bandshift, is that there is a region, a particular region, also a fairly long region, because it's 600 nucleotide long, in the 3' UTR of TDP-43 that was capable of binding itself. And as you can see here, this was validated by bandshift analysis. And the presence of this region was really responsible for the autoregulation of the protein, because we, I must say, very nicely demonstrated it, because we made a GFP reporter protein that only carried the last part of the 3' UTR, the 2 exons of TDP-43 and the entire 3' UTR sequence. And you can see that when we transfected this reporter construct into the cells that were capable of producing the wild-type transgene, if we just make a small deletion inside this TDP-43 binding region that we called TDP-BR, then autoregulation efficiently occurs after you switch on the expression of the transgene. But if you remove the entire 600 nucleotide region, then autoregulation does not occur anymore. And so at the end of or rather at the beginning of last year, what we ended up was publishing a preliminary model for the autoregulation of TDP-43, whereby normally TDP-43 is transcribed using two different polyadenylation, exploiting two different polyadenylation sites. If you have a lot of TDP-43 in the cell, then this TDP-43 will bind the TAR, the TDP-43 binding region, and somehow get rid of the messenger RNAs, therefore decreasing the amount of TDP-43 in the cell. And of course, you have the opposite situation. So if for whatever reason the levels of TDP-43 in the cells drop below a critical threshold, then there will be less available to bind this regulatory region here, and you will have more the production of more stable and better translated mRNA, TDP-43 mRNA isoforms, and automatically the cellular concentrations will increase. And in the last year or so, very briefly, what we did was to try and refine a little bit this model, so and see exactly what are the consequences of TDP-43 binding to this region. And of course, one of the major consequences is that if you have, and that is fairly intuitive, if you have a RNA polymerase II that is transcribing through the TDP-43 gene, and eventually you get to transcribe a sequence that is bound by a lot of TDP-43, then this RNA pol II will somehow get into a conflict with this protein, and therefore it can stall upon transcription. And this is something that we immediately tested using a particular kind of analysis that's called run-on analysis. So basically using a series of probes that you can put you can use on the endogenous, by looking at the endogenous pre-mRNA. What you can do is or the endogenous mRNA, what you can do is to use how well the RNA is transcribed in these different regions. And you can see that you have an increase of RNA production just at the beginning of the mRNA, because this is something that, because you have RNA-POL2 that is accumulating in the promoter region, and that is just starting to transcribe. Then as the RNA-POL2 starts to run along and starts transcribing, it gets diluted, and so you get to a situation where, for example, here in one of the middle exon of TDP43, you have a dilution of the newly synthesized mRNA. But when you get into very near the region of the 3' UTR where TDP43 is binding, you see that there is a stalling of the RNA-POL2, and you have a kind of slowing down of the RNA-POL2 due to the higher occupancy of this region of the RNA by TDP43. And what this is doing actually is doing several things. The first thing is that it will cause the splicing of an intron that normally is not recognized. So you see here through RT-PCR analysis, so you have the splicing of intron 7, the use of the polyadenylation one side is almost completely abolished, and what you start getting is the use of another polyadenylation site that is right next to it, and that is completely and that is nuclear retained, because what we also did was to actually look at the RNA differentiating between the nucleus and the cytoplasm, and you can see that whilst all the mRNAs that use polyadenylation 1, they are present both in the nucleus and the cytoplasm, whether you are in conditions of TDP43 overexpression or not. The polyadenylation 2 that appears only when you overexpress TDP43, and therefore you have a slowdown and an increased splicing of this intron 7, they are mostly retained in the nucleus and they're not present in the cytoplasm. So although this mRNA species could actually code for the TDP43 protein itself, it will not do so because it is mostly kept in the nucleus and therefore quite far away from the ribosome. And there is some read-through, but also in the case of read-through, so in the case when the intron 7 is not recognized and is not taken out, what happens is that there is competition, because TDP43 itself is not just capable of binding to the TARDP region that is somewhere around here, but is also capable of binding to the downstream sequence element of the polyadenylation 1 and inhibiting its use. So this is really a very efficient way that the cell uses when it has a lot of TDP43 to keep down the use of the poly-A1 site and favor the use of downstream sites that are not as efficient. TDP43 is mainly binding to the region here, TAR DNA binding protein. But as you see from the band shift analysis, the 3' UTR, yes. When you see the band shift analysis, you see that there are loose motifs all over this place here. And so this is the major 600 nucleotide region that is bound by TDP43. But you also have, by RIP chip analysis, you also have binding of TDP43 to the DSC element of poly-A1, and therefore inhibiting the recognition of the poly-A1 by the CSTF64 factor. And this is something, this this activation of intron 7 skipping is something that you can actually also see in vivo in the brain of mouse models, because now there are several mouse models that have been appearing in the literature where they overexpress TDP43. Sometimes in the wild type, but other times in a mutated form. And one of the things that we noticed is that in several of these mouse transgenic models where they overexpress TDP43, then the overexpression of the mutant form of TDP43 was accompanied by the downregulation of the endogenous mouse TDP43 production. This can be this is actually to be expected, because if you look at the conservation of the human and mouse TDP43 mRNAs, you see that in the 3' UTR region, the conservation is almost as good as in the exonic sequences. And indeed, if you look more closely at the different polyadenylation sites, you see that all of them, with the exception of polyadenylation site 3 that is never actually used also in humans, they are all well conserved in mouse. And if you analyze by simply RT-PCR the mouse brains to see what is going on as far as polyadenylation use is concerned, what you see is that also in this case there is normally there is the use of the polyadenylation site 1, just like in humans in normal conditions, but in the conditions where the transgene is expressed, then you start to see quite a prominent increase of poly-A2 of intron 7 skipping and use of the poly-A2 site that is nuclear retained. So in mice and in vivo, you see actually a situation that is very similar to what we saw in the cell lines. And this is just to give you an idea of just how important is this splicing of intron 7, and therefore the removal of poly-A1 from the 3' UTR of TDP43, because we made a very complex series of constructs where we deleted several regions and portions of the 3' UTR, but the bottom line is that if you remove several regions, you still get some degree of autoregulation. The only mutants in which you really fail to get any autoregulation whatsoever are the mutants where we actually remove the possibility of intron 7 splicing. So we simply mutate the splice sites and we remove the ability of poly-A1 to be physically removed from the 3' UTR. And this is actually confirmed by RT-PCR analysis, because you see here that, for example, a construct that does not autoregulate is the construct that do not autoregulate are the construct that always can use the poly-A1 site, whilst all the constructs that give a good or middle degree of autoregulation, they can actually start losing the ability to recognize the poly-A1 site because the intron 7 is removed, and they start to use less efficient polyadenylation sequences. And so this is really the model that we have now. So in normal conditions, the POL2, whilst it's transcribing TDP43, is just running nicely along the gene and transcribing the poly-A1 site that is chosen very efficiently. The ones that keep on going can manage to actually recognize another poly-A4 site that we are still investigating, and production goes on quite efficiently. If you have a lot of TDP43 that is binding to the TDP43 region, then the presence of this TDP43 causes a stalling of the polymerase in this position here, and this stalling can actually favor the excision of the intron 7 with the removal of the poly-A1, and the switch to a less efficient polyadenylation. And also you have even when this removal does not occur, the poly-A1 use is inhibited by direct competition with the CSTF64 sequence. And so in the last few minutes, the idea is to actually go back and see if you have disaggregation and you lose TDP43 presence in the nucleus, what happens to the splicing process. And we started by doing a very simple, in the beginning, we have several examples where TDP43 is playing a role. So CFTR is the one where we initially recognized it. A few years later, we found another example where TDP43 was binding to a UG repeat before APOE2, and it was also inhibiting recognition of the exon. And then more recently, there is another gene that was published by Don Cleveland and then later confirmed by Philip Kale, where the UG repeats, so the binding site for TDP43 was present near, not near the 3' splice sites like CFTR and APOE2, but near the 5' splice site. And in this case, TDP43 was acting as a splicing activator. So in parallel with the studies on autoregulation, what we did was to test if the presence of UG repeats near the 5' splice site and therefore TDP43 binding there was actually capable of affecting splicing. We therefore did a bioinformatic analysis to search for all the genes that had a UG repeats very near to the 3' splice site, and we tested if there were any changes in the recognition of the exons that carried UG repeats near the 5' splice site when we removed TDP43 from the cells. I must say that at the endogenous level, we did not manage to see any changes. So this means that basically having a UG sequence very near to the 5' splice site of an exon is not usually sufficient to either act as a TDP43 binding site or to be very capable of affecting the recognition of the exon. But when we inserted some of the most likely candidates, because they were identified as TDP43 targets in vivo, and we put the sequence of this exon with the UG repeats near the 5' splice site, then we managed to actually see that removing of the, we put these sequences in the minigene, and then we removed TDP43 and added back the SI-resistant wild-type or mutated TDP43 construct. What we saw is that in 3 cases, so in one case it was the BRCA1 gene that carried a disease-causing mutation. In this case, you see that normally you have the normal recognition of the BRCA1 exon 12, even when there is the disease-causing mutation in position +1, although you start gaining some exon skipping and cryptic splice site recognition. If you remove TDP43, then the proper recognition of the exon is completely absent, and of course you can restore it if you put back the wild type, and you cannot restore it if you put back a mutant that cannot bind RNA. So at least in this case, a UG repeat near the 5' splice site can, in theory, have an effect, and we found a few other of these examples in the for the ATF1 gene, where also near to the exon 7, there were these UG repeats, and when they were inserted in the minigene, actually exon 7, in this case, the skipping of exon 7, and we removed, in this case, TDP43 was acting as a negative regulator, because when we removed it, there was inclusion, and also when we put back the wild type, we recovered the skipping ability. So another one was the retinoid X receptor. And so the situation that we have now, and I think I will stop here because we're over time, the situation that we have now is that whilst at the 3' splice site of TDP43, TDP43 is mainly acting as a negative regulator, when TDP43 is binding near UG repeats to 5' splice site of different kind of exons, then in many cases it may have no effect at all, but in some cases it may act both as a negative or as a positive splicing factor. And this is it, and I'll take any questions.